My name is Joel Ong. I'm an associate professor in computational arts at York University. I'm also the director for Sensorium, the Center for Digital Arts and Technology there. So I am a media artist, and a lot of my work is inspired by the sciences. I have a background in biology. As an undergrad, I did the life sciences and ecology, and I have a degree in biological arts. I have a master's degree in biological arts from Symbiotica in Western Australia. So a lot of my art projects are focused on the intersections of art and science, and in particular understanding the world through multiple different lenses. I'm very interested in ways that we can expand our perspectival spaces, so be it through expanded sensory awarenesses of our environment, or through different modes of visualization and sonification, where scientific data can be more easily understood, or at least tell a story about how the world is. So the story with Untitled Interspecies Umwelten is an expansive project. It is an expansive project that has been on my mind, I think, for a really long time now, but only started to materialize just slightly after the lockdown. I realized that we were spending a lot of time in close proximity with microbes and microorganisms in the environment, and having access to a microscope, which I highly encourage everyone to do. Having access to a microscope allowed me to look at these little critters in much higher detail, and I started spending a lot more time with them. From there, I was thinking, how can we construct an interspecies communicative portal or develop a relationship where we truly begin to understand what's going on in our environment? And I think these are speculative, of course. There is the sense that every species has its own bubble of perception, and so we may never be able to enter that being human, and all we are left with are anthropocentric terms of references, like speech, like text, like language, even numbers, and all forms of, computational analyses. These are all very human and very anthropocentric. So it may not be, it may not be the most productive to think of an interspecies language as being centered around a human language. But for me, in this time, this milestone, I guess, in this project at OCAD, at On-Site Gallery, this is the project that's being developed here. So the project that I'm presenting at On-Site Gallery is called Untitled Interspecies Umwelten. And when you approach it, you will see a microscopic live feed of this single-celled algae called Euglena gracilis swimming around under the microscope. And you'll see these interesting little yellow contours that kind of frame its body as it's moving across the screen. And this is a result of a rudimentary computer vision algorithm that is tracking its shape, and it's letting you know how much it resembles a circle. In the research around Euglena gracilis, a lot of it is still kind of unknown, but the single-cell algae exhibits really intricate and very complex body contortions or movements, and that's what they call metaboly. And this metaboly is very closely related to the amount of stress it is experiencing. So if we think about it moving, kind of flat and elongated, it would be in a relatively happy state, excited state, or what I would like to term, what I would see as a curious state. And then if it's kind of sandwiched or smushed up into a ball, somewhat circular and a little bit pensive, then it would be the opposite of curious or excited. And so, the project basically takes a very simple confidence level of how much it resembles a circle and creates a gradient of what the euglena may speculatively be feeling, be it sad, much closely related to a circle, or excited, closely related to, or less likely, less resembling a circle. In addition to my piece, every piece in this exhibition really tries to engage with different perspectives of the world through the lenses of other species, other agents, through very innovative portals such as VR, visualizations, interactive exhibitions, just beautiful experiences for you to really come and sit down and listen to and experience the works. I think that it's very, very special to have this exhibition as a milestone because it really expands our perspectival. This exhibition really creates a perspectival shift in our understanding of interspecies co-relationships. So if we're living with and breathing with multiple different organisms in the environment already, how can we actually see them? How can we understand them? How can we create a narrative? Or how can we exhibit what has been called promiscuous care in the environment, where we care not only for our kin in our species boundaries, but also just moving across that to the microorganisms, for instance, or other organisms like plants, like forests, and in my case, algae, which you can't actually see nor touch. I started working with euglena about maybe 3 or 4 years ago. I was really inspired by research that was coming out of Boston and elsewhere about euglena as part of a system as part of a gaming system. So there was a game called Euglena Pac-Man that came out, which basically moved the euglena around a small space by turning on and off lights around a dish. And I found it was really interesting because much as it was a microorganism, that means that it's really small and you can only access it with these technologies like the microscope and other kinds of computer-aided technologies that I mentioned. Its movements were really in our time scale, within our time scale. So as it moves, you can develop direct feedback from it. And so these people these researchers who were working on the game project with euglena could see the euglena move across the screen really fast. They could move it around so much so that they could actually interact with it and have it play a game with them. And I was really interested in this relationship. I didn't think to make a game out of it, but spending time with the euglena itself made me think about its body movements as a form of dance. Its choreography, if you will, of its body's contortions and movements as a form of narrative. And so I've been working on this project for a while now, just to see how we can use this understanding, or how we can develop deeper this understanding of what it means when it does what it does. And what I will say is, what I've learned, I think, over the 3 years is that, much as scientific literature has said that they're very robust creatures and they survive under very, dire conditions, with very little water, without sunlight or without food, they are also very fragile. And I think this exhibition has shown, leaving them in a microfluidics chip over a duration of time. It's been out now for almost 3 weeks. It is starting to exhibit signs of stress, and so amounts of care will need to be taken place over the duration of this exhibition itself. And so the work isn't done when it's put up or when the opening is over. In fact, the work is a continuation over the duration of the exhibition.